first, a quick apology. I had the lecture notes all ready to go last night at 10, and I forgot to upload them. So they are there now as of about, 3 minutes ago, maybe? 4? So they are there. Another quick thing is a shameless plug for a talk which will be going on next Tuesday at noon over in Smith Center by a postdoctoral researcher in my research group, Viruses from Heaven and Hell, an Infectious History of Life. So great introduction to virology if you're planning on taking virology, and a lot of fun even if you're not planning on taking virology next term. Also, next Wednesday will be a guest lecture by a graduate student who's working in my lab on CRISPRs. And usually he has clicker questions. It'll be recorded, or sort of the same thing. And I will do my best to make sure the notes are posted beforehand. Yeah?
Is it exam material?
Is it exam material? Let me see. No, anything that's in lectures is going to be on exams. So how nitpicky I will be about it, that's another question. But yeah, I'm going to be sitting in the back taking notes. And actually, the exam questions are coming from my notes. He might come up with a question or two. I'll come up with a question or two. Right in the middle in the back. And they won't be questions. They'll just be notes. I'll use those notes to put my questions together later. So now the surprise for all of you. Today's lecture, which actually is not dissimilar from last time. And I think I finally sorted out the problem with all the recordings. It was this stupid USB hub that drops stuff. Go figure. So today we're going to continue talking about transcriptional regulation. And I should have had this outline for our last couple of lectures because we already talked about DNA binding, helix turn helix motifs, the methods about how to think about DNA binding of these specific DNA binding proteins, particularly the transcriptional regulators. And then in terms of bacteria, repressors, activators, and how those interact with each other. And then we talked a little bit about the activators last time as far as eukaryotes. And today we're going to continue and talk about some of the repressors and chromatin, and also a couple of examples of transcriptional regulation. And someone will ask me, can't think who it might be, is this going to be on an exam? So some of this stuff is very, very detailed, and I wouldn't ask you something very specifically detailed. Actually, I'm going to ask you some clicker questions, which are extremely detailed, but you've got the notes, you've got what I've just talked about. So that being said, I want to ask you very, very specific questions about how one particular activator interacts in one place. Much more interested in concepts, and all those concepts really have to do with combinatorial control. And when I say combinatorial control, I really mean different regulators interacting on one gene. And the examples I'm going to use, the reason I'm using these examples is because they are examples of combinatorial control. So you've got activators, you've got repressors, how those interact with each other in a particular cell at a particular time is going to determine what kind of readout you have after that. Regulatory circuits also, again, connected to that whole sort of idea. And then we'll probably talk a little bit about epigenetics and DNA methylation. That'll depend on how far we get. So this next slide is actually kind of a good review, if it's actually going to work, which is apparently not. So much for my great ideas on how I get this, sorted all these things out. Unplug this. Now it's not responding at all. How nice. Move forward with this one. Try this method instead. Yay! Go figure. So yeah, how we get regulators and how you regulate transcription at eukaryotic promoters. So it usually has to do with some kind of interaction with these sequence-specific gene regulatory proteins. So here you have a gene regulatory protein which is bound to the DNA, helps and binds another transcriptional regulator, and that other transcriptional regulator will help with this whole cascade of processes that eventually leads to transcription, which is what we have down here at the bottom. That's one case. Another pretty general case is that you have these transcriptional regulators that will interact in some way with all of these proteins that are involved in the pre-initiation complex. So they could be the general transcription factors, TF2D, TF2B, they could be the polymerase. In most cases, they're actually going to be the mediator complex, which is associated with all of these. And all that does is it gets the RNA polymerase and binds it to the promoter. So getting the RNA polymerase to the promoter, and okay, what's missing here? What's missing in all of these DNAs? Eukaryotic DNAs. What eukaryotic DNAs bound by? It's Friday morning, it's snowing, it's cold. Nucleosomes, chromatin. So that's one of the big problems here, getting everything to the promoter. This big, humongous complex. Getting a big, humongous complex to a particular place on DNA is really hard in eukaryotic systems because you have all of this chromatin around. So getting all of that together in one place, this is probably the majority of transcriptional regulation, is getting enough activators together to get the polymerase bound to the promoter. In a few cases, and this is actually the case that I studied during my PhD, again looking at a bacterial system, is you have binding by a transcriptional regulator, and then it allows the already bound transcription complex to start making its messenger RNA. And then in the last case, and this is actually probably the rarest case, the RNA polymerase has actually started to move, started to transcribe, but it gets stuck. And that stuck is a pause, that's what we call it, and it's just not going any further. Then it needs the activity of this kind of activator in order to get it to move. So these are the main processes, particularly this one right here, which is where you get the polymerase to the promoter. That's probably the most critical of any of these. So, but of course I mentioned what's missing here. What's missing here are all of the nucleosomes. So how do you get a pre-initiation complex, which is the RNA polymerase and all these general transcription factors, to a promoter? And that promoter is just outlined here basically as being the TATA box. And so in this case, the TATA box is not accessible to all of the components. So what happens is you have some kind of activator protein that will bind to this sequence. And again, there's a double bar here, which means this could be long distance away. This is a classic enhancer. So enhancer binding protein, that's this green protein. In many cases, that's going to interact with some kind of modifying enzyme for histones. And the classic modification enzyme is a histone acetyltransferase. It puts acetyl groups on histone H3 or any of these different histones, but particularly histone H3. And that particular modification is also being done by something, we already talked about these coactivators before. So it's a coactivator. Coactivators by definition are transcriptional activators but don't bind to DNA by themselves. So they have to be brought in by protein-protein interactions with some other sequence-specific DNA binding protein. So this kind of modification, sometimes you'll have more modifications as well that take place. In this case, it's a phosphorylation. That's not as critical. You have even more modifications that happen to all of these different tails. And then TF2D will recognize all of these modified histones and allow the pre-initiation complex to start. And so it's not so much about the DNA binding interactions here, it's really about the protein-protein interactions. So protein-protein interactions between activators and coactivators, protein-protein interactions between TF2D and modified histones, and then finally you can start to assemble the rest of the pre-initiation complex. But that's really the major thing which is going on here. And I put up bromodomains. What the heck is a bromodomain? So a bromodomain it's a domain of a particular protein that interacts with acetylated lysines. So bromodomains here, again, are interacting with these acetylated lysines. Bromodomains, there are a number of proteins in TF2D that have these bromodomains, which helps to bring things to promoters and literally just bringing things to acetylated histones. There's also something called a chromodomain, and those chromodomains interact with sort of the flip side, and I like to think this is sort of the heterochromatin domain. Those guys are interacting with the methylated histone side chains. So bromodomains, acetylated side chains, chromodomains, methylated side chains. So yeah, this process, again, but it's DNA binding by a sequence-specific DNA binding protein, and then the protein-protein interactions, which eventually will get the whole pre-initiation complex to assemble. And then once that's happened, you're good to go. So at the very beginning, I should back up here real quickly. We had this example of one activator binding to, again, mostly through protein-protein interactions, and bringing in a second activator. Why is this so useful? Well, this is actually so useful because people have looked at things like the reporter genes that we talked about last time, where you had mixing and matching of different domains. Here, if you have binding by one transcriptional activator, you have a small amount of transcription. And again, this is all from studying these kinds of reporter genes. If you have binding by a different transcriptional activator, you'll have a different amount of transcriptional activation. But when both of them bind together, you end up with much, much more transcription, and much more than just an additive amount between these two. And so the idea here is that really the binding of these multiple proteins is a synergetic process. And so this is, example 1 + 2 = 100. Here, 1 unit, 2 units, 100 units. But this is, again, it's going to vary, but it's really all about the constellation of these different proteins which are going to interact with all the other things which are being assembled at the start site, at your site of initiation. And so what that means is it's this combinatorial process. And we could, if you're really interested in the details on why this works, it has to do with binding coefficients and dissociation constants of all the different proteins. But we're not going to get into that. So this is, again, just sort of yet another overview of the process. What's happening first? You have a sequence-specific DNA binding protein that binds to chromatin. Most cases, you'll have a chromatin remodeling complex as well because, again, these are big complexes, and nucleosome-bound templates do not work very well. A lot of that has to do with histone modification enzymes, and these guys can really be simultaneous and relative to each other. But these kinds of chromatin remodeling and histone modifications, these seem to happen at almost all of the genes which are going to be activated. We also looked last time. Most of the time you're going to have more than one activator protein that all come together and eventually get your RNA polymerase bound to the promoter, and then once it's bound, you can start to get transcriptional initiation. But it's really about usually these first couple of steps that you have to have before you end up with finally getting all of your RNA polymerase together. And this is, again, multiple different steps, and it's different for different genes, and we're still trying to work out how it works for all kinds of different genes. And then when George, my graduate student, mentions and talks about some of the RNA next Wednesday, there are also a bunch of RNAs that are involved in this process, so it's not just proteins as well. Questions, comments, worries? Ready for clicker questions? Yes, we're all ready for clicker questions. Good. Okay. So, and you don't have your notes to refer back to, or at least some of you won't. Find somebody who does. Which of the following seems to be the first step in most eukaryotic transcriptional activation events? Enhancer binding protein binds to enhancer, covalent histone modification, mediator DNA binding, RNA polymerase DNA binding, or TF2H activation? We haven't hit 100% yet, have we? Except for the one time when I showed you the answers already, before I turned stuff off. 5, 2, 1. Consensus we do not have. So let's try about it. See if we can get 100% this time. Okay. Ready to go again? Yes, no?
Are enhancers the same thing as activators?
Pardon?
Are enhancers the same thing as activators?
Ask your neighbor.
Yes.
My DNA is collapse one of these days. Ten, 5, okay, we seem to have a slight difference of opinion here. We're not at 100% yet. Enhancer binding proteins. What's an enhancer? What's an enhancer? So, an enhancer is a DNA sequence that an activator of transcription binds to, by definition, because it's enhancing transcription. We'll talk about silencers as well. I'm surprised nobody asked me what a silencer was. Silencers do exactly the same thing. They bind to repressors, and they will stop transcription. Histone modification, this has to happen in most cases, but it's usually dependent on first the binding of one of these activators. Mediator DNA binding, mediator doesn't bind to DNA. RNA polymerase DNA binding, that does have to happen eventually, but it's rare that that's happening at the very first step because everything's bound to chromatin, and the polymerase is big, much bigger than any of these transcriptional regulators. And nobody liked the activation of TF2H, but neither do I. So that makes 100 and something of us, 106 of us. Okay, good. So I want to talk about the other aspect. So again, repressors. You have repressors just like you have activators. In eukaryotic systems, you have to have many more activators. In bacterial systems, you have to have many more repressors in terms of regulation. But you also have repressors working in eukaryotic systems. So repressors can work in multiple different ways. The sort of classic way that repressors work is a lot like the way that we talked about repressors working in bacteria. They block the binding of something else. And they can block, actually, the binding of the general transcription factors and the polymerase. But more frequently, what seems to happen is they're binding to a particular DNA binding site that overlaps with the DNA binding site of an activator. And we'll see some examples of this a little bit later on. So this binding by the repressor doesn't allow the activator to bind, as we just talked about in the clickers. That's the first step that you need to activate one particular gene. So no binding of activator, no activation of that particular gene. We also last time talked about how these activators work. They're modular. They've got DNA binding domains. They've got activation domains. These activation domains are sticky. They bind to other proteins and bring them into the pre-initiation complex. Well, if the repressor blocks that sticky piece, then the activator can't do its job anymore. So it's another way that activators work. And you can have binding sites for repressors that are going to overlap with your promoter, or they're going to interact directly with the general transcription factors and basically hold on to them or block their interactions with, say, the RNA polymerase. These are what I like to call direct acting repressors, because they're doing something that is either directly interacting with an activator, directly interacting with some component of the general transcription complex. On the other hand, we also have indirect repressors. So indirect repressors are going to act not in terms of specifically interacting with activators or the general transcription complex. They're going to interact with chromatin. And so they're interacting with chromatin in some way to basically shut down the chromatin so that the polymerase can't get to the promoter. So some of these, and anything now is red, these are the repressors, the ones in red, are now going to interact with chromatin remodeling complexes that will block binding to a promoter, again symbolized here by the Tata box. You also have repressors that interact through protein-protein interactions with histone deacetylases. So if we said a histone acetyltransferase is a coactivator, what will be a histone deacetylase? A co-repressor, exactly. So a lot of the co-repressors are histone deacetylases or histone methyltransferases, because methylating the histones doesn't allow them then to be acetylated later on, and also helps binding by these chromodomain containing proteins, which contribute to the formation of heterochromatin. So shutting down the chromatin, not allowing the transcriptional complex to bind here at your promoter. So we have activators, we have repressors. They can act directly through protein-protein interactions with the transcription machinery, or they can be working indirectly through modification of chromatin. Okay, so let's look at a couple of examples of these things. These are just 5 different examples. Again, I'm not going to ask you exactly which ones are which, or if I do, I'll give you a lot of detail in any particular exam question. But positional expression in Drosophila embryos. Drosophila embryos are great because you can see the whole cell. It's got multiple nuclei in it, and the regulation of genes in any given nuclei you can actually observe quite well. People have been studying Drosophila genetics for a long period of time. Yeast mating type determination. Yeast, again, nice model organism that there's really good genetics for. And a lot of what we have learned about chromatin modifications, how particular genes act together, comes from studying this yeast mating type. Now, yeast are really wonderful as model organisms because they can function both as haploids and diploids. And it turns out that how a yeast knows, to totally over-anthropomorphize, that it's haploid or diploid or one of the 2 different mating types, which are A and alpha, has to do with the activity of transcriptional regulators, both activators and repressors. Actually this is slightly out of order here, talk a little bit about some eye development again in fruit flies, because again these are wonderful model organisms, lots of really good genetics here, and then talk about some more general aspects through the switch in lambda. We talked a little bit about lambda before in terms of integration. There's a site-specific recombinase that takes the lambda genome and puts it into the host genome, but there has to be some decision that's made before that, whether to integrate or whether not to integrate. And that decision-making process also has to do with the presence of different transcriptional regulators. Eye development happens to be basically it's one master regulator, and we talked a little bit about the master regulators before. We'll talk about them again. Induced pluripotent stem cells. So just a couple of transcriptional activators can lead to all kinds of developmental processes, and that's because they turn on other transcriptional activators. So if one transcriptional activator turns on a second transcriptional activator, which can turn on a third transcriptional activator, et cetera, that gives a whole cascade which can generate things like eyes, for instance. So how can you get, particularly if you're thinking about multiple cells, different genes being expressed in different parts of some kind of multicellular organism? This is just an example of how to do this very conceptually. If you start with a single cell which then divides, there's some kind of signal on the left and on the right. On the right-hand side, you turn on gene 1, transcriptional regulator. And you just have this sort of same rule that you keep going through here, left versus right in terms of each of these cell divisions. So here, the next round of division, all the cells that are on the left get 2, all the cells that are on the right get 3. But these are descended from the cells that are on the right in the first place. So these now have 1 and 2 or 1 and 3. And the same thing can go on just using exactly these very, very simple rules in terms of left versus right, in terms of getting completely different transcriptional regulators present in different cells in different parts of a developing embryo. And the best studied of these are the Drosophila embryos. And Drosophila embryos have a very clear, at least if you're a Drosophila geneticist, front end and a back end. So here's your front end, here's your back end. These are actually determined based on the egg structure. And so what the mother or the maternal processes that make up an egg actually determine what's the front end of the egg and the back end of the egg. And the front end of the egg is going to turn into a head. The back end of the egg is going to turn into a tail. And that's all due to these kinds of cell division processes and having a signal at one end versus the other end. And it turns out these signals are all due to RNA that we'll talk about later. Probably one of the most striking of these is if you look at expression of one particular gene. And this is what's called the even-skipped gene. So if you look at a Drosophila embryo, like this, the even-skip gene is expressed in these 7 stripes throughout the embryo. And what's important here is not so much that they're expressed, and you can have a gene that's expressed everywhere, but that they're also not expressed in certain stripes. And before we get into more detail about this, one of the things which is really important about these Drosophila embryos is they actually have multiple nuclei, and these are all nuclei really spread on the outside of the embryo, and no cells. So these are all just a multinucleate cell, one ginormous cell that has all these different nuclei. And these different nuclei are expressing different proteins depending on different places where they are in the cell. If you go and look at where this gene is in the genome, because we have the Drosophila genome, we figure out where all these genes are. The even-skip gene has a nice promoter sequence, and then it's got a whole bunch of regulatory sequences upstream of the transcriptional start site. And what people found is, it turns out that each of these regulatory segments seems to determine expression in one of these stripes in a different place in the embryo. The reason we know this is you can take one of these regulatory segments and put it into a completely separate piece of DNA that has a reporter gene attached to it, the LAC-Z gene, again, beta-galactosidase. It's great because you can take a colorless dye, and in the presence of this enzyme, it will chop it and make it turn blue. So you take this reporter gene fusion, again, fusion is because it's got this element and the reporter gene, put into all of the nuclei in this embryo, and then look and see where the LAC-Z gene is being transcribed and then translated and made into protein. It turns out it's just in one stripe, and it corresponds exactly to where this stripe in even-skipped is. And you can do the same thing with these other regulatory elements. You find out that it's expressed in all kinds of other different stripes in different places. So how can this be happening? How can you have gene expression just in this one particular place in the embryo and not in all of these other places? Well, that turns out to be due to the presence of just 4 gene regulatory proteins. And for most genes, there are way more than 4 regulatory proteins. And there are actually more than 4 regulatory proteins that are involved in regulation of the other stripes. But if you just look at the gene regulatory region for stripe 2, it's dependent on 4 different transcriptional regulators. Bicoid and hunchback, which, as you'll see, are close to the front end, or the anterior end of the embryo. And then a couple of repressor proteins that are also present in stripes. And the reason they're present in stripes is actually a lot like why the even-skipped, and particularly even-skipped stripe 2 are in one particular place as well. So we'll not worry about why that is. But we've got these 4 different proteins. And these 4 different proteins are going to determine why you get expression in just stripe 2 of one of these reporter genes when you have that particular gene regulatory sequence. So what's in this gene regulatory sequence? The gene regulatory sequence is up here in gray. And in that gene regulatory sequence, you have binding sites for all of these 4 different proteins. So we've got bicoid and hunchback. They're down here, this sort of yellow piece and the blue piece. And then we have cripple and giant, which are here on the top. Oh, by the way, I forget if we talked about this already. The Drosophila geneticists love to name their genes based on the phenotype of the mutant. And so if you're missing giant, the Drosophila doesn't have giant, it makes giant embryos. If you're missing hunchback, it makes a hunchback-looking embryo. If you're missing bicoid, it actually ends up with 2 ends that look the same. And anyone here know enough German to tell me what cripple is? Cripple is a cripple. Actually, it's the same root. How do you know that an embryo is crippled? I have no idea. And probably not politically correct either. So, but yeah, these are the binding sites. And so literally, you can just take this piece of DNA, do those kinds of experiments that we talked about before, band shift experiments, footprinting experiments, etc. And what you see is that there's a lot of overlapping binding sites. So these repressors right here overlap with where activator binding is. A couple of places where you've got activators and repressors which are separate. And again, activators overlapping with repressors. So it's just this combination of these 4 genes and this regulatory sequence which gives expression of just stripe 2. So how does that work? The way that works is that you have these gradients, and again, bicoid and hunchback are the activators, giant and cripple are the repressors. And if you look carefully at exactly this region right here, right at the end of where you have expression of your bicoid and expression of hunchback, again, these are the activators. You have to have both activators present in order to get activation of the reporter gene in this particular stripe. If you have either of these repressor genes here, giant or cripple, you're not expressing. And so this yellow region right here in the middle, that's where you have expression. That's because you have the activators all bound because there are no repressors around. On this side, you don't have expression because giant is around. On this side, you don't have expression because cripple is around. So it's literally just the position of where you have your repressors, where you have your activators, which can give you expression in this one particular line. The other thing is that all of the other places in the Drosophila embryo, even skipped is off. If you add all the other transcriptional regulators, then you end up with the 7 stripes that we saw way back here. Here we are, the 7 different stripes. But if we just think about this stripe right here, that's due to those 4 regulators interacting with this particular regulatory element. So combination repressors and activators at that particular position. So everybody got this? You've taken good notes? Yes? No? If you're investigating Eve expression in a Drosophila embryo that's missing the giant gene but is otherwise normal, you would expect normal Eve expression lacking in stripe 2 but normal in all other stripes, additional Eve expression posterior to stripe 2, additional Eve expression anterior to stripe 2, or no Eve expression whatsoever. And I have a sneaking suspicion we may need a while for this. And this is the kind of question I would not ask on an exam because this would be too specific. it definitely looks like we're going to need some more time for this. If this, would it help to have this up for a second? Yes, I thought it might. So again, so you're missing giant, but otherwise normal. So what would you expect? Normal, lacking in stripe 2, but everywhere else. Posterior, additional anterior, or no expression whatsoever. Okay, so again, this is the embryo that's lacking giant, doesn't have the giant gene in it, and therefore would also not have the giant protein. Okay, we ready to go again? Yes, no, maybe? Okay, take a look at the clicker question again. So missing giant, otherwise normal, normal Eve, lacking in stripe 2 but normal in all the other stripes, or additional expression posterior to stripe 2, additional anterior, or no Eve expression. So okay, I'll back up again so people can once again look at this. So. Ten, 5, 2, 1, stop. Okay, what do people think? Well, most people think this. Do I agree with you? So let's back up and take a quick look at this. So if we're missing the giant gene, what does giant do? It's a repressor. So you're missing a repressor. You're missing the repressor. That means you're going to have activators present here anterior to stripe 2. So downstream, or so I should say downstream of this, posterior, you still got the cripple regulator. So it's not going to be there. And lacking in stripe 2, it's going to be expressed just fine in stripe 2 because you still have these two activators which are present here in stripe 2. Yes, no, happy, less happy? Okay, so D is correct, yes. But interestingly enough, beforehand, most people had elected C. So I guess discussions help at least a little bit. Okay, so this is sort of that overview again. We've talked about the activators, repressors. You have combinations of these proteins interacting with everything at the top of the box. These can be positively regulating. They can be negatively regulating. All of these things are working together to try and give you a particular probability that you're going to get transcriptional initiation. And this is, again, that example we just talked about is a really simple example of how gene regulation is working. For most genes, it's multiple different proteins, multiple different DNA binding proteins, co-activators, co-repressors, etc. And it's going to be exactly the constellation that you need at a particular time with a particular signal, which is going to lead to initiation of transcription or not. This is partly why it's really hard to predict what's going on with some of these things. And in fact, one just example of this is here. We have a nice example of a heterodimeric basic helix leucine zipper protein. The names here are not important. No, I'm not going to ask you them on an exam. But I think it's a nice example of where we have a heterodimer, again, leucine zipper with a basic helix. This basic helix is fitting into major grooves right here in DNA. And another protein, this is another activator protein, IRF4, which if I remember correctly is a homeodomain containing protein. And this is an alpha helix in a major groove right next to it. Turns out that both of these interactions still lead to expression of different genes in T cells and cause them to develop into other kinds of T cells. This is just, again, it's an example of this combinatorial regulation, in this case, 3 different proteins all acting together to give you activation. Fortunately, there are a few genes that seem to work way better than others, and in terms of transcriptional activation. And these are what people call master regulators. And again, this is work that's mostly been done in Drosophila. Here we have a slightly more developed Drosophila embryo, anterior to posterior. There's a whole group of cells that normally will give you eye structures. Turns out that there's a protein which is being expressed here, which is not being expressed elsewhere in the embryo. And you can literally take that regulator called eyeless, again, Drosophila geneticists. They name the mutant after, their name of the gene after the mutant phenotype. So you get rid of this gene, you have no eyes. But it turns out you put this gene back in a different place in the embryo, it can actually make eyes on legs or other particular places in the embryo. So it's literally where this master regulator is being expressed can give you this particular structure. Turns out that there's not enough nerves that attach to it, so this eye can't actually see, but it still develops all of the appropriate cell types in order to give you eyes in just really weird places. There's another fascinating regulatory protein called antennapedia. And what that does is it actually will transform antenna into legs. And so these flies actually have legs growing out of their heads with the expression of actually lacking this antennapedia gene. So few genes, and it turns out Drosophila is a little simpler than our system, so the single regulator genes don't seem to have quite as strong an effect as they do in Drosophila. But we did talk last time about these induced pluripotent stem cells, and this is one of the things that I love or hate, probably depends on your point of view. Hate, I would say, probably if you're a student trying to learn these things. You remember that you needed 4 activators, right, to get you an induced pluripotent stem cell. This slide, of course, has 3. I told you last time sometimes you can have 2. We had a slide that had 6 last time. So which ones are they? Which ones are we supposed to remember for the exam? The message here, and I haven't written the exam, so I don't know yet, but I doubt I'm going to ask you the names of these things, but that you need very few genes to be overexpressed in a particular cell type to give it this induced pluripotent stem cell phenotype. I think that's reasonable. Whether it's 1 or 3 or 5 or 4, that I don't think is particularly reasonable. And the names of them, I can't remember. It's OCT4, SOX2, and I thought it was KIF and not KLF4 and NANOG or whatever. But there's very few of these master regulator proteins that can then turn an otherwise differentiated cell into a dedifferentiated cell that can then differentiate into a whole bunch of other cells. And again, this is really particularly exciting for thinking about personalized medicine, getting cells from a patient, modifying those, and then being able to use those cells to treat neural disease or something like that. So although, to be perfectly honest, there's a number of companies that say that their stem cells are terribly wonderful, and a lot of the treatments actually are garbage. So be careful about some of these things, and the jury's still out, I think, on how good some of this is actually going to be for personalized medicine processes. One of these sort of master regulator proteins, which has been studied extremely well, also by people who are authors of this textbook that we're using, so partly why it's used so much in the textbook, is something called the glucocorticoid receptor. And the glucocorticoid receptor is actually a really nice example of how you could have a signal lead to a transcriptional output. The normal ligand for the glucocorticoid receptor is cortisol, classic hormone, and actually it turns out there are lots of different hormones here that can bind to these glucocorticoid receptors, and it turns out there are lots of different hormone receptors. What happens with these hormone receptors is they're usually in the cytoplasm of the cell. They're present in the cytoplasm of the cell. They only will be transported to the nucleus once you have the presence of one of these hormones. So the presence of one of these hormones, then the glucocorticoid receptor, or any of these hormone receptors, goes into the nucleus, binds to DNA, and can cause great upregulation of a number of genes. But it's really dependent, just like we had with the lambda repressor before, where this particular binding site is for the glucocorticoid receptor, whether it's going to act as a repressor or an activator. And how it works in any particular cell is going to depend on the other proteins that are present in that cell. And so this is, to start cartoon-wise, if you've got a brown protein, a blue protein, and this orange and yellow protein, that's going to be present in one particular cell. So the glucocorticoid receptor is going to turn on these three different genes. But say in a different cell, maybe you don't have this brown protein there because it's a different part of the organism. It wasn't next to the anterior or the posterior, when you're setting up your axis. And so you'll be expressing different genes. So it's all about the combination of the different genes which are there. This glucocorticoid receptor is present in almost all cells, but how it's acting is going to depend on the presence of all of these other kinds of proteins. Now, it turns out the glucocorticoid receptor is a little too complicated to get into as a particular example. So now we'll talk about a much simpler one, and when you're talking about simple systems in eukaryotes, usually you end up talking about yeast. So yeast, they're great because they can replicate as haploids, they can replicate as diploids, but they're undergoing sexual reproduction because you've got the haploids that come together to form a diploid. So what are the sexes of yeast? Do you have male yeast and female yeast? No. You just have A yeast and alpha yeast. And how do you know they're A and alpha yeast? Because when you get two of them together, they do something called schmooing. And this is the terminology, is the shmoo. So what happens is you get the two together, they actually start to grow towards each other. And so the shmoo that happens, which eventually will cause cell fusion to take place, and you end up with a diploid. But this only happens if you have an A cell and an alpha cell. If you have two A cells, they're no schmooing with you. Or two alpha cells, same thing. So it's got to be an A cell and an alpha cell to interact to give you a diploid and get these schmoos and eventually move on to this process. But if you just look at the yeast, the yeast know that they're alpha or A, but a researcher really has no idea what they are. So it turns out that these yeasts are actually genetically almost identical to each other, the A's and the alphas. The difference turns out to be a very small part of the genome, only around about 1,000 nucleotides, called the MAT locus, so the mating type locus. And the sequences which are present at the mating type locus are going to determine whether your yeast is alpha or your yeast is A. But otherwise they're identical. And what's even more weird is that if you take yeast and just grow them, they can switch. I say, I'm bored with being A, I want to be alpha. Or, I'm bored with being alpha, I want to be A. And that's not quite true. It actually turns out it goes from generation to generation. They will actually switch going from one to the other. So how can you switch having a sequence that determines whether you're A or a sequence that determines whether you're alpha? And these sequences have to come from somewhere. And so what, again, to make a long story short, it turns out that there are copies of alpha and A genes at different parts of the chromosome in all yeast, in A yeasts, in alpha yeasts, etc. These are in silent parts of the genome. So a lot of what we know about silencing and heterochromatin actually comes from studying A's and alphas and whether those genes are being expressed or not. So this process of going from alpha to A and A to alpha, again, has been very, very well studied. And a lot of what we know about chromatin and how chromatin modification happens has to do with this study of looking at yeast mating type. And so the previous one was a figure from our textbook. This is from a slightly different textbook. But this is the basic idea. Is you have the genetic information for alpha at one end of a chromosome. You have the genetic information for A at the other end of the chromosome. These are close to telomeres, are in silenced regions of the genome. And they're silenced because there's heterochromatin here. And that heterochromatin is blocked from spreading by some of these barrier proteins, again, discovered in yeast, so that this just stays as heterochromatin. Now, somehow you've got this extra copy here at the mat locus. This is now active. This doesn't have silences associated. It's a nice piece of euchromatin. So how do you get this, actually literally a change from alpha to A and A to alpha? It turns out that this process is actually a lot like we have with homologous recombination. There's an endonuclease that will cut here in the mat locus and use the information at one of these flanking loci to copy it into this locus. So if you're alpha, you cut alpha with this endonuclease, that gets replaced with A. If your A gets cut by the endonuclease, it gets replaced by alpha. And these sequences, again, over here are completely silenced. But when they're at the mat locus, there actually are three genes which are made from here. Alpha 1, which is made from these A sequences, A1 made from the A sequences, and alpha 1 and alpha 2, which are made from the alpha sequences. And it turns out that these are the only proteins that are going to give you one that likes to shmoo or one that doesn't like to shmoo, and also has haploid-specific genes versus diploid-specific genes. So how does that done? Turns out it's just, again, these three proteins together with some other proteins that are always present inside the cell, and that, in fact, turns out in this case just one protein, MCM1. So A locus, we have MAT-A1, and in the cell, we have MCM1. Alpha locus, MAT-alpha1, MAT-alpha2, and it's the combination of these proteins that will give you an A cell or an alpha cell. A cells, it turns out, are turned on because they've got this MCM1 protein bound to A-specific genes. Nothing interacts with alpha-specific genes, so those are off. Haploid-specific genes are on because of this sequence right here. If you're an alpha cell, then you make MAT-alpha1 and MAT-alpha2. MAT-alpha2 binds to MCM1 and blocks as a co-repressor alpha-specific, A-specific genes. MAT-alpha1 will turn on alpha-specific genes and nothing interacts with these haploid-specific genes. So if you've got one cell that's got A, one cell that's got alpha, these guys shmoo, and you end up with a diploid, which is an A alpha diploid. The A locus, again, so it's diploids. You've got two copies now, one of A and one of alpha. A is going to continue to make MAD-A1. Alpha is going to make MAD-alpha-2. It's actually not going to make MAD-alpha-1. Why does it not make MAD-alpha-1? Because MAD-alpha-1 and MAD-A1 are really good repressors. They repress the haploid-specific genes. They also repress MAD-alpha-1. MAD-alpha-1 would give you expression of your alpha-specific genes. No MAD-alpha-1, no alpha-specific genes. MAD-alpha-2, together with MCM1, turns off the A-specific genes. So now you have none of these genes on because of the combination of MAD-A1 and MAD-alpha-2. So we've all got this down well enough we can answer a clicker question on it, right? Okay, I'll move forward and then we can move back. So in yeast lacking MAD-A1, see, I love these mutant forms, in a diploid cell, you would expect haploid-specific genes to be expressed, alpha-specific genes, A-specific genes, or some combination thereof. Okay, so we can think about this. I'll make our decisions. If we still got that, do you want me to back up quickly? Okay, so lacking MAD-A1, what are you going to expect in a diploid cell? Lacking A1, what do you see in a diploid-specific cell? Okay. It looks like a lot of people would like some more time. And we're nowhere near a consensus. So let's go back and take a look at this again. So lacking MAD-A1 in a diploid cell, what do you expect? Haploid alpha or A's or some combination thereof. Again, lacking A1, diploid cells. Yep. Okay, let's go. 10, 5, 1, 0. Okay, we seem to have difference of opinion here. Either just haploid or haploid and A-specific genes. So let's back up and take a quick look. So in the absence of A1, okay, you're not going to be repressing the haploid-specific genes. You're definitely getting haploid-specific genes. The question is, are you also getting expression of A-specific genes or alpha-specific genes? So alpha-specific genes are turned on by MAT alpha 1. Why are you not getting MAT alpha 1 here? Because MAT A1 and MAT alpha 2 are suppressing MAT alpha 1. So what does that mean about alpha-specific genes? They're turned on, right? So alpha-specific genes and haploid-specific genes. So D it is. This competition here seems to be a little rough. Yeah. So the question was here, really briefly, before everybody runs away, which is probably what we'll do because it's Friday, is specificity. Am I likely to ask a question like this, like exactly like that on the exam without a lot more context? No. The first question, however, I think that's totally fair game to have something like that on a final. So, which I forget exactly what it was, but skim back to that. So next time we will talk about phage lambda and the lambda genetic switch because viruses are too exciting not to spend enough time on. And as again a quick reminder for those of you who are interested, on Tuesday, but also tomorrow, we've got Ladies Lab Night, the Biology Graduate Student Symposium, and Mario Capecci.